Hello, I am Anmol CK, first MSc Biology, St. Mary's College, Thrissur. Today we are discussing about the topic methods of identification of microorganisms, morphological analysis and biochemical tests. Microbes. The major groups of organisms studied in microbiology are bacteria, algae, fungi, viruses, and protozoa. All are widely distributed in nature. Most microbes consist of a single cell. Microbes range in size from small viruses 20 nm in diameter to large protozoa 5 mm or more in diameter. Bacteria. The majority of bacteria are single-celled organisms with spherical, rod or spiral shapes, but a few types form filaments. Bacteria are cellular, they do not have a cell nucleus. Many bacteria absorb nutrients from their environment, but some make their own nutrients by photosynthesis or other synthetic process. Archaea. Very similar to bacteria are the group known as archaea, and the bacteria belong in the same kingdom called Monera. Like bacteria, the archaea are single-celled and do not have a nucleus. Algae. Many algae are single-celled microscopic organisms, but some marine algae are large, relatively complex multicellular organisms. Algae have a clearly defined cell nucleus and numerous membrane-enclosed intracellular structures. Fungi. Like algae, many fungi such as yeasts and some molds are single-celled microscopic organisms. Fungi also have a cell nucleus and intracellular structures, widely distributed in water and soil as decomposers of dead organisms. Viruses. Viruses are acellular entities too small to be seen with a light microscope, composed of a specific chemical substances, a nucleic acid and a few proteins. Viruses replicate themselves. Many viruses can invade human cells and cause diseases. Protozoa. Protozoa are also single-celled microscopic organisms with at least one nucleus and numerous intracellular structures. Most protozoa can move, but a few, especially those that cause human disease, cannot. Found in water and soil environment. Next is morphology. A primary means of identifying bacteria is by their morphology. Example: shape, size, and aggregation properties when viewed in the light microscope. First one is cell shape. Bacteria exhibit three main steps which are determined by the molecular properties of the cell wall. These include rod-like forms, spherical forms, spiral and curved forms. In addition, few species termed pleomorphic lack of rigid cell wall and thus have a no defined shape. Next is size. Most bacteria are 1 to 5 microns in length. Thread-like species can be many times longer than their cross-sectional diameter. Typically, a bacterium is some 100-fold larger than a virus and then 10-fold smaller than a eukaryotic cell. Next one is aggregation properties. Whereas the single isolated cell is the fundamental bacterial unit, many species exhibit distinctive aggregation states. For example, long chains, irregular clusters, or regular clusters resembling crystals. These aggregation states are determined by their orientation of the cell division plane to the axis of the cell and also the tendency of progeny cells to adhere to each other. The aggregates are also relatively stable and characteristic of the species can be disrupted mechanically without loss of cell viability. Next is biochemical tests. First one is indole test. Microorganisms can metabolize tryptophan by the tryptophanase. It is based on the formation of hydrolytic product from amino acid tryptophan. Media, tryptophan used as a substrate. Reagent, Kovacs reagent. Color pink and red. Example, Protease mirabilis can differentiate from Protease vulgaris. Next test is citrate. This test is used to determine the ability of an organism using the citrate enzyme to use citric as its sole carbon source. Reagent, bromothiamol blue indicator. Positive reaction is growth with color change from green to intense along the line. Example, Salmonella, Citrobacter, Edwardsia, etc. Negative reaction is no growth and no color change, line remains green. Example, Escherichia, Shigella, Morganella, etc. Next test is triple sugar ion agar test. This is to differentiate among and between the members of Enterobacteriaceae family, used to determine carbohydrate fermentation and H2S production in bacteria. Interpretation: alkaline slant or acidic bud gives only glucose is fermented. Acidic slant or acidic bud representing glucose, sucrose and lactose, all three sugars are fermented. Bubbles or crack present represent gas production and black precipitate present is H2S production. Reference. Thank you.